I'd like to introduce our speaker today. Her name is Kristin Parent. She is an assistant professor who joined the Department of Biochemistry and Molecular Biology in the spring of 2013. She's one of our most promising young faculty. Her research focus is on understanding how viruses enter cells. So how does a virus gain access to a cell in your body? And there's been a recent breakthrough in the technology we use to actually visualize or image small molecules and biological systems like that. It's called cryo-electron microscopy, and she'll explain how it works. It's really quite a beautiful technology. Through this research, we now better understand how viruses find, attach to, and infect specific cellular hosts. In addition, her research has shed light on viral diversity in the environment, so the different types of viruses that one finds in the environment. Kristin has won a number of awards and grants for her work, for teaching and for mentoring students. She received both the MSU Outstanding Mentor Award and the MSU Teacher Scholar Award. For her research, Kristin has been awarded the AAAS Marion Milligan Mason Award for Early Stage Career Women in the Chemical Sciences, as well as the Young Investigator Award from the American Society for Microbiology. She's also recently been awarded an NSF, National Science Foundation, Career Award for her work on virus-host interaction, and in part for her role in involving undergraduate, high school, and middle school students in scientific research. So, Karen?
Good morning. I'm going to tell you about some research that we're doing in my lab on the study of viruses. And no doubt everybody in this room has probably had an intimate experience with a virus, probably not very pleasant at some point in their lives. From the common cold to flu to some of the emerging viruses that we're hearing about in the news, such as the Ebola outbreak a few years ago and more recently Zika virus, epidemics are a global problem, and one of the things that's really important to understand about viruses is the biological scale. There's a lot of them. So what I wanted to start out today was a clicker slide that I use in my introductory biology class to try to get students to think about the scale of biology. And I won't make anyone answer this question. I'll give you the answer, and you can impress your friends at parties this weekend. But it turns out that there's more than 10 to the 31 viruses in the biosphere. So if we write that out, you get an idea of the gargantuan size of this number. Practically speaking, what this means is if you went up to Lake Michigan and grabbed 2 scoopfuls of water in your hands, you would be holding more viral particles than there are humans on Earth. They have a highly diverse lifestyle, and they exist in many different all the kingdoms of life, and basically if there's a host out there, there's a virus that can infect it. So we as scientists can't possibly study all of these. So what we do is we study a class of viruses that tells us something about how more similar ones work. So the specific goals of my lab are to understand a type of virus called a bacteriophage that infects the gut microbiome. So things like Salmonella, E. coli, Shigella, which are in our guts. And what I want to understand is how viruses can infect these cells and how change occurs during virus evolution. And the way that we do this, at least in my lab, is to look at the structure of how these viruses attach to their cells. And this is a key theme in biology, that structure relates to how something works. So from these common household items on the slides, you can see very clearly that the way they're shaped is really important for the way that they work. Well, the same is true with molecules, but it gets to be a little difficult when you have to understand how to look at these molecules that are very, very tiny and too small to see. So for example, if anyone's had the common cold, and if you took a coin quarter, you can fit over 1 million of those particles from end to end of the quarter. So they're very, very tiny, and they're everywhere. So the technology we use to image them is something called cryo-electron microscopy, and this gained a lot of attention recently with the 2017 Nobel Prize being awarded to Jacques Dubochet, Joachim Frank, and Richard Henderson for their technological advances that helped this field. And recently, as you heard about, the technology improvements are basically due to 3 main things: better instruments, better cameras to record our data, and better computation. How this basically works is if you take a sample of virus, we have to get it into the microscope in a manner in which we can image it. So one thing we do is attach it to tiny little grids that are about 3 millimeters, blot away excess liquid, rapidly plunge freeze it into liquid ethane, which freezes the sample so fast that ice doesn't form, and what we get is a thin layer of viral particles. We can then look at them in different positions. When the electron beam hits them, we see a three-dimensional object make a 2D image. And you can see if you follow the tail of this bacteriophage, they exist in different positions. So we do this for hundreds of thousands, sometimes millions of particles, and through a long series of computational pipeline, we're able to get a three-dimensional structure. I'm going to skip the nitty-gritty of the computation, but I'm going to show you what the end product looks like. And this is a zoom-in view of a virus. This has over 700 individual proteins that we can model and understand how these proteins come together. And this is important because if we want to devise effective therapies such as drugs or antibody binding to viruses to help prevent infection, molecular detail at this level is very important. Another way that we can image the viruses is by looking at individual particles infecting biological membranes. And this is really important if we're able to utilize viruses to do good things. And that might seem a little contradictory if you've ever been sick with a virus, but it turns out they're really effective in dealing with bacterial infections. So there's increasing antibiotic resistance, and in patients with chronic infections, these multi-drug resistant bacteria become a huge problem, such as this patient who had cystic fibrosis. So my lab and many other labs around the country have been teaming together to find viruses in the environment that can infect these bacteria to help treat the clinical infections and to study the structures of how they interact with their hosts so that we can learn how to better replace antibiotics as is becoming increasingly necessary. So this is an example of a student-based project where an undergraduate in my lab, Will Dean, is on our microscope here. And this is actually a bacteriophage that we isolated as part of a class at MSU's campus from the Red Cedar River. So as part of the NSF Career Award, we're implementing this as part of the Biosciences Initiative into the introductory biology classes so that all first-year students and second-year students taking introductory biology can have an opportunity to participate in student-centered research. Furthermore, we have a new microscope has been recently purchased, and a microscope facility on campus is going to allow us to do some of the more modern computation and high throughput imaging. And we're hiring new faculty to build in this area, not only for virus-related research, but other biological work that can be done using cryo-electron microscopy. And so I'm trying to keep this brief. So at the end, I'm just going to say thank you to the people in my lab who have helped pioneer this work. My student Jason, Haley, Will, Madeline, Tori, Sarah, and John are great. And of course, thank you to the bacteria and the phage for letting us study them.
Very interesting.
I'm happy to take any questions.
Questions from anybody? It's almost hard to ask a question. This is so unbelievable, those numbers.
I've got a question for you. Just the cost of a microscope like that, what does that cost?
There's a range in instruments. The low-end scale is about $1 million, and the best one on the market is about $7 million right now. The one that we purchased is towards the higher end, yeah, about $3 million.
Is this I mean, some of your undergraduates, are they getting fired up when they hear this?
Oh yeah, they love it.
Yeah.
The high school students love it too. We're working with high school students around the country. We're starting in Nebraska right now to try to compare viruses that are in very different environments. And the high school students are getting very excited about the possibility of going to college to do research. So it's qualitatively through student feedback we've heard a great response.
Is there an online aspect where you could go, I mean, a unit that you could put out there for some, I don't know, biology classes or science classes that,
Yep.
that
Yeah, so if
I don't
you,
know if we do that.
if you go to my website,
I'm
yeah.
curious.
No, one of the things we have on our website are video protocols on how to do all this so that undergraduate researchers at other universities can learn how to do this in their classrooms. And I have to say, I'm not the first person to come up with this idea of phage hunting. This is initially made with the C-phages program through Howard Hughes Medical Institute. And we're simply adapting it to work here and in a different classroom setting. So there are many available protocols online.
We had some issues with the chemistry department, their audit, but in their protocols, do you have because some of these viruses, I suppose, could be, some kind of wild and crazy, dangerous virus. Do you have a set of protocols that are pretty severe
We do.
that you have to follow that?
The ones that the student use are the bacteriophage that infect non-pathogenic bacteria. So they're completely safe to work with in the benchtop, and even the middle school students can do this. Because we're sampling from local rivers and things like that, it's perfectly safe. I mean, people swim in these places.
Does some of yours get a little more edgy than you get out there a little bit?
My postdocs and grad students do work with some of the non-pathogenic, the pathogenic bacteria, but we do have biosafety protocols in place in the lab. Yeah.
Very good. Thank you so much. That's wonderful.